Okay. Good evening, everyone from India, and good morning to everyone from US. So it's a great honor to introduce today's speaker, Dr. Patrícia Ribeiro Pereira. She is an assistant professor at the Department of Radiology, Washington University School of Medicine. Before that, she had completed her Ph.D. in medicinal chemistry, and she carried out her postdoctoral research and was a research associate in the Department of Radiology at Memorial Sloan Kettering Cancer Center. She was mentored by Dr. Jason Lewis in oncologic molecular imaging. She applied radiolabeled antibodies and small molecules to determine biological mechanisms by which some patients do not respond to and also develop resistance to cancer therapies. Patrícia's research program at Manenkrot Institute of Radiology centers on tumor-targeted multimodal imaging techniques in concert with complementary genetic and proteomic approaches to investigate how regulation of membrane receptors can function in tumors and their microenvironment at the most basic level and at the level of drug resistance. Dr. Pereira is actively involved in the World Molecular Imaging Society and the Society of Nuclear Medicine and Molecular Imaging. And today she is going to talk to us about imaging tumor biology with radio-labeled antibodies. And for all those young students who have joined this webinar, I'm sure this is going to be a very knowledge-filled 1 hour. And I'm going to hand over the stage to Dr. Patrícia. And we will have a talk for about 45 minutes for all those audience. Please hold your questions to the end, and we will have an interactive session at the end.
All right.
Over to you, Dr. Patrícia.
Thank you so much. Thank you for the kind introduction and for the invitation, and to everyone who is joining today. So I'm a new PI here at WashU in St. Louis. I've been here since a little bit over 1.5 years. So I started my lab right after the lockdown, let's say. So it was a little challenging, but with the support that I have here, especially from Dr. Wall, our chair, things run smoothly, and I'm very excited to be talking to you about my program here at WashU for the next 45 minutes. So here we go. I have no disclosures for this presentation, and I like to start with this slide, tumor heterogeneity, because this is the motivation for most of the things we do in the lab. When we think about tumor heterogeneity, we think about comparing a primary tumor with metastasis. Most of the time, they are very, very different. Inside of the same tumor, the cells tend to group as clusters, and each one of these clusters comes with different levels of heterogeneity. And then even at the single cell level, tumors are also very heterogeneous. When we think about this, we think from comparing primary tumors with metastasis, where some of the clones develop over time, others might disappear during treatment, same thing. So it's very complex, as I'm sure you all know. And so we need approaches where we can visualize this heterogeneity and this complexity. And in my lab, we use molecular imaging to address some of these, especially positron emission tomography, with the idea of moving forward towards personalized medicine, where patients might need to have a specific treatment according to the disease that they have. Problem here is most of the time tumors or patients are selected for certain therapies based on analysis collected from tumor biopsies. Those come with limitations because we can collect all the tumor lesions, so we're having sampling bias. Challenge also to perform repeated biopsies. It always requires interpretation from a pathologist, and there is this complex biology that I'm gonna be talking to you in the next slides, and also the tumor heterogeneity that I mentioned. But there is a technique that we believe can help, and that technique is positron emission tomography that allows us to have images of the full body at different time points, allowing for a dynamic visualization. And we pair the image resolution of positron emission tomography with antibodies that have high specificity for certain targets in the tumors. And so we pair them with these positron-emitting radionuclides, and in that way we'll be delivering the antibody to the tumor site, allowing specific visualization of biological processes in the tumor. This is really exciting because if we look at the number of antibodies that have been approved in the last years, it's very impressive. And it's expected that this number will be higher for the years to come. So we can, what we do in the lab is basically we repurpose these antibodies that they are already FDA approved for therapy. We repurpose them for imaging. So they basically are the same thing that is already used for therapy, but just paired with a positron emitter. And when developing these antibodies for PET imaging, there are a couple of things to consider. I won't go into detail about all of those, but basically we don't want to disturb the antibody. We want to keep the binding domain intact so that the antibody still binds to the target in the tumor. We always need to consider which linker we're going to use to attach the antibody to a chelator, which chelator to use, and then the radiometal. So for the radiometal, we have different options, as I will show you in the next slides. And that option will always depend on the kind of antibody that we are using. And because of the technology that now we have, we can go from a full-length antibody, these IgGs, they have the light chains, heavy chains, or we can go to really smaller molecules like scFv, or only the binding domains are retained. So we go from things that they are 150 kilodaltons to really small biomolecules of 25 kilodaltons. They have advantages and disadvantages when we compare these different biomolecules. So in terms of an intact antibody, they are the ones with the higher residence time in the tumor site. In terms of the smaller molecules, they have a higher blood clearance when you compare with a full-length antibody, higher tumor penetration because they are really these small molecules, but they will have this high renal uptake. And so here just an example of a PET image with an intact antibody. Because they are large molecules, we often need to wait a few days, in this case 168 hours. Mini bodies, the tumors can be imaged at early time points such as 21 hours, and diabodies as early as 4 hours. From a patient perspective, maybe the smaller molecule will be better because the patient can receive this PET tracer and be imaged in the same day. But these intact antibodies still have advantage in the sense of this higher image resolution and high tumor uptake. And these full-length antibodies are what I will be focusing for today's talk. And as I said, we have different isotopes that we can use, and this will always depend on the biomolecule that we are using. So for a full-length antibody, as you can see here, it will take a few days to get to the tumor. So in that case, we want to use an isotope that has a long half-life, such as zirconium-89. So we always need to consider, we need to match the biological half-life of the biomolecule with the half-life of the radioisotope. But for smaller molecules, maybe we can use things like F18, which have half-life of 108 minutes, or gallium-68, copper-64 for the mini bodies, for example. So just a few things to consider when creating an antibody PET tracer. And with these things, we can image multiple things in the cells. What we most explore in my lab are membrane receptors, and the conjugation that we use, the labeling that we use most of the time is a full-length antibody, where we attach a chelator such as DFO to the lysine groups of the antibody, and then we use zirconium-89, which has this long half-life, to radiolabel full-length antibodies. And one of the membrane targets that I've been explored in the last years is the human epidermal growth factor receptor 2, abbreviated as HER2. So HER2 is actually expressed in both normal cells and also in cancer. The difference is that it is amplified or overexpressed in some tumor types. The most well-known is breast cancer. It's a pretty well-known target and validated therapeutic target in breast cancer, but also in gastric cancer. Even though these are not the only tumor types where HER2 is expressed, it's actually expressed in different tumor types, and it has been more and more studied as a potential therapeutic target for those tumor types, especially now with the newest antibody drug conjugates, such as the DS8201 that we'll be talking in the next slide. One thing with HER2, which is common for most of the membrane targets in cancer, is this heterogeneity. So in the same tumor tissue, we might have regions that they are pretty high in HER2, and right close to those, we might have regions that they have very low expression of HER2. And here's another example. Regions with high HER2 expression close to regions with very low levels. And so when a pathologist is looking at these and giving a score, so 3+ is for the high expression, 1+ is for the low expression, it might be quite challenging to really define what is a HER2 positive or a HER2 low or HER2 negative tumor. PET imaging can help because we have antibodies that bind HER2, such as trastuzumab. We can pair them with a PET tracer, and here's just an example preclinical study. This is a HER2 plus breast cancer preclinical model, BT474, and what we can see after injection is that there is some tumor accumulation as early as 10 hours, but still some antibody in circulation, and then over time the antibody is cleared and mostly accumulated in the tumor tissue. And here an example of a clinical image, again trastuzumab PET showing that we can detect multiple lesions in a person with metastasis. Other things that, in this case, trastuzumab PET has allowed is, for example, in this case, this is a woman with HER2 negative primary breast cancer. So by looking at the IHC for HER2, it was classified as HER2 negative. So this would be someone that wouldn't enter into HER2 target therapy, but this person received a PET scan with trastuzumab PET, and some lesions started to highlight. These were metastases, and these metastases were actually HER2 plus, HER2 positive. So this person entered into therapy with trastuzumab, and when we compare B and D in this image, we can see that some of the lesions really respond to the therapy. So this would be something that IHC would have missed, and trastuzumab PET allowed to detect this tumor and to select the best therapy for this person. Other things is in the context of monitoring tumor response, here a case of HER2 plus gastric cancer, where trastuzumab PET before therapy enables detection of multiple lesions in a person with gastric cancer. During therapy, we can see that some lesions are responding, like this one here that disappears. But over time, on progression, we can see some new lesions, like this one in here on the left, and the one in the middle that is just has higher signal compared with what it was at the beginning. What was really cool in this study was also that a good correlation was found between PET and HER2 copy number of copies for HER2 that code for the HER2 protein. So showing that PET imaging, trastuzumab PET, can allow monitoring tumor response, but also correlates well with HER2 levels in these tumors. Another thing that some of these trials have shown is, for example, in the Zephyr clinical trial, this was a trial with TDM1, which is trastuzumab with a chemo drug. And what that trial showed was that although HER2 is present, the tumors express HER2, for some reason, these tumors did not show uptake for trastuzumab. So HER2 is there, but for some reason, the antibody is not binding these tumors. And there are a number of biological processes that could explain this. So maybe the levels of HER2 were low for the antibody to bind to it. Maybe there was saturation of HER2, internalization, or even other kinetic features such as perfusion and vascularization. In my lab, what we look into is whether or not the dynamics of these receptors, the dynamics of the HER2 receptor, could explain why some of these tumors have HER2, but there is no uptake on the antibody. And that is because the receptors, they are not always available on the surface. They actually get internalized through endocytic pathways where they can be degraded or they can recycle back to the cell surface. Now, when we use these antibody therapeutics, we really need these receptors to be on the surface. Antibodies are pretty big molecules, and they need to bind to the receptor to then be able to internalize into the tumor cell and exert their therapeutic effect. And amongst these, there are several different endocytic pathways. One of the pathways that we explore the most in the lab is a caveolin-mediated endocytic pathway. These caveolae are these structures on the cell surface, and they are rich in caveolin-1. It's their main structural protein, but also in cholesterol. And initially, when starting to look into whether or not HER2 could be internalized by caveolae and what would be the role of this endocytic pathway for this receptor, we started looking into gastric tumors from preclinical models. And what we started to see was the tumors grouping these different clusters, as we were expecting, different levels of heterogeneity in terms of HER2 expression. But what was interesting was when we look into clusters, for example, here 1 and 2, we can see these are clusters that have more HER2 staining on the surface. And these clusters have low levels of the caveolin-1 protein. Clusters 3 and 4, they have low cell surface staining for the HER2, and they have high expression of caveolin-1. So suggesting there was an inverse relationship between these two in terms of caveolin-1 protein levels and HER2 staining on the surface. We did several mechanistic studies with the knockdown, CRISPR amplification, and we were able to validate this inverse relationship. What we also did was we implanted mice with patient-derived xenografts. So these are tumors collected from a patient, again here, gastric cancer, implanted into a mouse. And what we saw again was that they show different levels in terms of HER2 staining on the surface, although they are classified as high expressors for HER2 based on IHC. And for example, PDXs 1 and 2, they had a very diffuse HER2 staining, and these were tumors with high levels of caveolin-1. PDXs 3 to 6, they had more HER2 staining on the surface, and they had low levels of the caveolin-1 protein. So again, showing in a more clinically relevant setting that there is this inverse relationship between these two proteins. We're also able to validate an antibody for caveolin-1 IHC that now we use in our approaches to classify high expressors for caveolin-1 and low expressors for caveolin-1. And by doing that and by dividing our tumor models into caveolin-1 high versus caveolin-1 low, again, they are all HER2 expressors. We can see the first thing is that the ones that have high caveolin-1, they have low uptake for this drug, the TDM1 drug, when compared with the ones that have low levels of caveolin-1 protein. And from here, we thought, okay, all of our data is indicating that these two are inversely related. So we did several in vitro studies showing that when we deplete caveolin-1 protein, we have more of the HER2 on the surface. We have more uptake for these antibodies to bind to the tumors. And from here, we wanted to use a pharmacologic approach where we could deplete caveolin-1, but that could have potential for translation. And the approach that we decided to use was using cholesterol depleting drugs, because as I mentioned at the beginning, these caveolae structures, they are rich in cholesterol and also in caveolin-1. And what we started to see was the use of cholesterol depleting drugs such as lovastatin. These are drugs that they are prescribed all over the world for people with high cholesterol levels. But what we started to see was when we use the statins, we have depletion of the caveolin 1 without alterations in HER2 signaling. But what was really fascinating was this is a study in mice, so a preclinical study. We had cohorts of saline mice treated with a saline solution or treated with a statin. And then we collected these tumors. And what we saw was the ones treated with the statin really had higher staining for the HER2 when compared with the saline tumors, showing that maybe now we have more chance for the antibody to bind to these receptors. And to validate this, that's when we bring in PET imaging. So again, using antibodies that target HER2, such as trastuzumab and pertuzumab, paired with the long-lived isotope zirconium-89. When we look at the saline cohorts, we can see tumor uptake of 8 hours, still antibody in circulation. Then the antibody clears from the blood, and we start to see a delineation of the tumors. Same thing, similarly for pertuzumab. Now, in the statin treated cohorts, we are able now to delineate tumors as early as 4 hours, and by 48 hours, the accumulation of the antibody in the tumor site is higher when in the statin groups when compared with the saline groups. So suggesting that by using a statin to enhance HER2 on the surface, we are enhancing accumulation of these antibodies in the tumors, so we are able to deliver more of these drugs to these tumors. And we're also able to validate these in our PDXs. So what was interesting here was that in these PDXs, only the ones that had high levels of CAV1 showed an improvement in accumulation when they were treated with the statin. The ones that had low levels of CAV1 really showed no major differences, suggesting that this approach could work on these CAV1 high tumors. We did several randomized preclinical studies testing the combination of these statins with drugs such as trastuzumab or T-DM1. And what we saw was here, when we look at the red line, the trastuzumab statin cohorts have a higher mouse survival when compared with trastuzumab alone in the green line, and same thing, or even more impressive, for T-DM1 combined with statin. T-DM1 again is a trastuzumab conjugated with the chemo drug, and again we are able to highly improve mouse survival when compared with the T-DM1 drug alone. And we also have been able to validate all of these preclinical data in retrospective clinical data, since these are drugs that sometimes patients are taking because of their high cholesterol when they are being treated for cancer. So we have been able to validate these more retrospectively in gastric and also in breast cancer. And now for the next slides, I will focus more on these antibody drug conjugates. So this is a market that is really growing and it will continue to grow in the next years. These are antibodies that they are conjugated with chemotherapeutics, and for HER2, we have the trastuzumab emtansine or T-DM1, and we also have trastuzumab deruxtecan, which is a drug that people are really excited about because it has shown response in both HER2 positive and also HER2 low tumors. And here the idea with imaging is to use an antibody with a PET tracer to image these tumors, treat them with an antibody drug conjugate, and then image them again with PET. So this is basically the same antibody it will be used for imaging and also to treat the patient. It just we switch the PET tracer by the chemotherapeutic drug. Here's just an example comparing FDG with copper-64 trastuzumab. And we can see that in responder tumors, we can see tumors disappearing on follow-up treatment with TDM1 on both on the FDG, and they were also delineated with copper-64 trastuzumab. While in the non-responders, we can see that copper-64 trastuzumab is able to delineate tumors, and on FDG, we see that these tumors really do not respond to this TDM1 drug. These drugs are very effective. They are, again, antibodies conjugated with chemotherapeutic drugs, but they also come with some side effects. And here's just another example on how PET imaging with the zirconium-89 trastuzumab can help monitoring patients that show failure to TDM1 therapy, where again, a negative pattern in the PET scan or low signal relates with failure to these therapies. But as I was saying, these antibody drug conjugates, they come with some side effects. And so one of the things we are exploring is by using these pharmacologic approaches, such as the statin, where we were able to enhance antibody accumulation in the tumors, could we use these to reduce the number of cycles that we need to achieve therapy with an antibody drug conjugate? And so that's what we did here in this study. This is a study with control mice, mice that we treat with a single dose of TDM1. We can see some, the tumors don't grow too fast at the beginning, but then a little before day 20, they start to grow. When we combine them with the statin, we can see that there is a difference in their growth. And when we compare either a multiple cycle dose schedule or a single dose schedule, we can see that the single dose works as effectively as the multiple dose when combined with the statin. But what we also see is that after day 30 or so, these tumors regrow. And so then we start to think, what can we do here in cases where, okay, maybe a single dose is feasible if we combine them with the statin, but what can we do after day 30 for these tumors that they start to regrow? And there was a very interesting study published 3 years ago or so now. And in that study, what was shown was that tyrosine kinase inhibitors, such as neratinib, could be used to enhance the internalization of antibody drug conjugates. So what we thought in our approach was, okay, we know that the statins are able to have, enable for more HER2s to be on the surface of the cell. So when we administer an antibody drug conjugate, we'll have more of those bound to the surface of the tumor cell. Could we then combine these with a tyrosine kinase inhibitor, such as neratinib, based on that study published in 2020, where if on the first steps would be enhancing accumulation on the surface, then would be enhancing drug internalization. And so that's what we did here. We had different cohorts, and we treated these mice with the antibody drug conjugate alone, in combination with neratinib or statin, or in combination with both of these drugs. And what we see here now, and this is still an ongoing study at this point, is still pilot data, but what we can see now is when now we try this triple combination of TDM1 combined with neratinib and statin, we are now able to really, we have a high difference between those tumors, or the ones that they were treated with TDM1 combined with the statin, or combined with the neratinib alone. And we also used, in this case, PET imaging, not with combining zirconium-89, but copper-64, where we labeled trastuzumab with copper-64. And we could use PET imaging to show high uptake of the antibody in the tumors pre-therapy. So they are HER2 expressors, they have high uptake. And post-therapy with this triple combination of TDM1, neratinib, and statin, we have a high decreasing signal. And we're also able to validate our PET images with expression levels at the tumor site, where we show that both total HER2, but also the phosphorylation of this receptor are decreased when we use this combination. Another antibody drug conjugate that we are exploring is the trastuzumab deruxtecan. As I told you, this is an antibody drug conjugate that people are really excited about. So we are exploring also this antibody drug conjugate in our studies and trying to see whether or not PET imaging could be used to annotate for changes in tumor volume and also in HER2 expression in these tumors. So this is a mouse image before therapy with the trastuzumab deruxtecan, and we can see high signal. They are HER2 expressors. We see that some mice, although they are implanted with the same cell line, some of them respond, like these ones here at the bottom, but others do not respond. And so then when we do a PET scan, we can see that the non-responders, maybe they have a slightly higher uptake, so they are still expressors, while the non-responders might not. And so we are still exploring a little more into what's happening in these tumors and why are some of them responding while the others are not responding, and what can we do about the non-responders so we reverse this drug resistance on them. And another thing that we explore in the lab is in the context of radiotheranostics, and using, the same antibody paired with the PET tracers such as zirconium-89 or copper-64, and then switching the tracer. So instead of an imaging isotope, we use a therapeutic isotope such as lutetium 177, copper-67. So here's just an example. In the case of conjugating trastuzumab with zirconium-89, we can image tumors. But we can switch the tracer now to lutetium 177, and we use that to treat. So it's the same antibody, but just conjugated with different isotopes, and we can either image or treat the tumor. And in the context of this work with the statins here, just to show that also the statins are able to enhance the accumulation of these trastuzumab labeled with 177 lutetium, where again, the statin-treated tumors have higher uptake when compared with the controls. And then when these mice are treated with 177 lutetium trastuzumab, they show response, but the mouse survival is higher in the statin cohort when compared with the trastuzumab alone cohort. And in this context of using these antibodies labeled with a therapeutic isotope, as I said, antibodies have a longer circulation half-life, so that can lead to the lutetium 177 to be delivered to non-tumor tissues. And also, because of those of that long circulation half-life, it's not feasible to image patients on the same day. Most of the time, patients do not live close to the hospital. So what can we do about this? So we can use the antibody fragments that I showed you at the beginning, or we can use a pre-targeting approach. And this is really exciting, especially now that, the latest Nobel Prize in chemistry was in click chemistry approaches, which is what bases this pre-targeted approach in here. There is a challenge in here, which is with the internalizing receptors, as I will show you in the next slides. But basically, in this pre-targeted approach, the way it works is there is a first step where an antibody conjugated with a TCO molecule is injected. This antibody at this point is not radioactive. It's just conjugated with TCO. The antibody will be in circulation, and then it will accumulate at the tumor site. After that, when we have this maximum accumulation in the tumor site, we inject a small molecule that has a tetrazine moiety. And this is the molecule that is labeled with the isotope, either an imaging isotope or a therapeutic isotope. Because it's a small molecule, it will clear fast. And the reaction between this TCO and tetrazine is a click chemistry based reaction. And so the tetrazine will click with these TCOs on the antibodies on the tumor site, but it will also clear faster in terms of the tetrazine that didn't bind to the tumors. And so we'll have less of this radiation going to the non-tumor tissues. But if you look into this scheme, it's all based on the hypothesis that by the time that the small molecule conjugated with the tetrazine gets to the tumor, the antibodies with TCO are still on the surface. And this is a problem with internalizing receptors, because when the antibodies bind to them on the surface, most of the time they internalize. And so what we thought was, okay, we have a pharmacologic approach where we can enhance the receptors on the surface, such as HER2, which is a highly internalizing receptor. Could we use this to allow for a pre-targeted approach in these receptors? And so here what we do is we give a statin before the antibody. So the statin will allow for more HER2s available on the surface. We inject then the antibody with the TCO, which will bind HER2s on the surface of the cell, and then we inject the small molecule tetrazine. There is this in vivo click reaction between the small molecule and the antibody, and then there is rapid clearance of the unbound small molecules tetrazines. And so we are exploring this in the lab. We did some initial studies to show that maybe this is feasible, but it's still not optimal because, well, as you can see here, this is a saline versus a statin-treated mouse, and we can see more tumor accumulation in the statin-treated mouse with this approach, but we still have accumulation in other non-tumor tissues, and still needs to optimize these time points between injection of the two and doses of the statin that will allow for this approach to work. While exploring these widely prescribed drugs, such as lovastatin, most recently, we were exploring other widely prescribed drugs, such as anti-diabetic drugs, and in this case, metformin. And this was really surprising to us. This is a Western blot in cancer cells. And what was surprising in here was that cancer cells treated for only 2 hours with metformin, which is an anti-diabetic drug, we see a high decrease in membrane HER2 accompanied by internalization of this receptor. And so we decided to do a PET study to see whether or not we could validate these Western blots. And again, as compared with the control, these metformin-treated mice show really lower signal when compared with the control. So that brings in a lot of questions. One of them is, let's say there is a woman with breast cancer, HER2 plus breast cancer, that is being treated with trastuzumab. It also has diabetes and is taking metformin. So maybe if this person gets a PET scan, maybe it will show up as a false negative because the metformin in there is enhancing the internalization of this HER2 receptor. And so it will show up on the PET scan as a HER2 low when it should be a HER2 high. There is also questions about what does it mean in terms of therapy. And so we are exploring this in the lab. Another question we had is this a temporal effect? And we can answer that question using PET imaging with copper 64. And so what we did in here was we performed a PET scan before treating mice with metformin, and we see accumulation in the tumor. During metformin treatment, we see a reduction in of the trastuzumab uptake on these tumors. Then we remove metformin from these mice for 1 week, and what we see is a rebound on the receptors, which is accompanied by a rebound on the PET signals, showing that yes, this is temporal, and maybe if a patient stops metformin before getting a PET scan, maybe will show up as as it should as a HER2 plus on the PET scan. So these are still some of the things we are exploring as part of this study. Now for the last slides of the talk, one of the receptors I want to talk about is something more related with immunotherapy, related with PD-L1. And again, here, in terms of the conjugation and labeling, it's almost the same as the one I described before for HER2. And so in terms of the immunotherapeutics, we have drugs that target both PD1, which is a cell surface protein. It's expressed in T cells. We also have drugs that target PD-L1. PD-L1 is expressed on the tumor cells. And in my lab, we focus mostly on receptors in the tumors. And so we are exploring more of the PD-L1 receptor. In immunotherapies, they work, but they don't work for all the tumors. And even in preclinical models. So these are MC38 cancer cells inoculated in mice, and these are CT26s. They have PD-L1 expression. When these are treated with a PD-L1 drug, we see some of them are non-responders and some of them are responders, although they have PD-L1 expression. And so one of the questions we have is, why do some of these tumors respond to these therapies while others do not respond? And even in the clinical setting, we have a high number of patients that had no objective response. Roughly 70% of them, they still do not respond to these therapies. And so why is that? And the second question is, what can we do about the ones that they do not respond? And the PD-L1 is quite challenging as a receptor, not like the HER2 receptor. Here are some of the things why it's challenging. First of all, it's not only expressed on the tumors. We have PD-L1 expression on tumors and in the stroma. Here is shown in this IHC image. The other thing is mainly expressed on immune cells, so it won't be mostly expressed on the tumor. It will be mainly expressed on the immune cells. Another challenge, these are antibodies tested in clinical samples for IHC of PD-L1, and they all show different readouts. So maybe these two at the top will show us PD-L1 negative tumors, the ones at the bottom will show us PD-L1 positive tumors. And the other thing is the dynamics of these receptor. It's a very, very dynamic receptor, and here just showing how it behaves on tumor cells or immune cells. Again, tumor cells, there is slight increase in PD-L1 after tumors are inoculated, but this increase is higher in the immune cells. And also it changes a lot during therapy. So there is this dynamic expression, this complex biology on this receptor, which makes it challenging, but also exciting. And since there are so many drugs targeting this receptor, we better understand more of the biology of PD-L1. This is one study showing how we can use imaging to visualize some of these immune targets, such as PD-L1 and PD-1, and how it compares with FDG. On PD-L1, we have, different organs lined up. We also see the tumor. PD-1, same thing. And in that study, the authors also showed that the responders, the tumors that respond to these immunotherapies, they have high uptake for PD-L1 and PD-1 when compared with the non-responders. So maybe PET imaging can help in defining response to these immunotherapeutic drugs. Another study where it was shown that the uptake of, this is a PD-L1 antibody atezolizumab, showing that the uptake correlates with response. So the ones that showed complete response to this immunotherapeutic, they showed higher uptake on the PET scan. So showing that maybe PET imaging can also be used to monitor response. And another thing that study showed was, again, highlighting the heterogeneity of PD-L1 and showing a correlation between PET imaging or uptake of this atezolizumab with IHC on the tumors, where this region here has high uptake based on autoradiography, and it has high expression for PD-L1, and these low regions have a low uptake but have high expression of PD-L1, again showing this heterogeneity and the complexity for PD-L1. In my lab, we are exploring how endocytosis relates to some of this heterogeneity. In these preclinical models, we see uptake in the tumor. We also see uptake in spleen, lymph nodes, which is common for these antibodies. We see this high heterogeneity on PD-L1, which in our model seems to co-localize with levels of caveolin-1 in the tumors. In the meantime, we started to explore more and more how caveolin-mediated endocytosis relates to PD-L1, and we developed a caveolin-1 inducible, a teton-inducible model of caveolin-1 knockdown, where we can knock down protein in vivo in these mice. And what we saw through PET was that there is a high increase in antibody accumulation in these tumors. And so we are still exploring more of this relationship in here. And so just a conclusion, because I think it's finishing. So I do believe antibody PETs can really provide several complementary information to some of the information we get through tumor biopsies. I hope I was able to show you that this technology allows us to monitor heterogeneity, but also the dynamics of membrane receptors in tumors. We're also developing these methods where we can modulate membrane trafficking and increasing receptor availability on the surface in a way that we enhance response to antibody therapies, and more in the context of theranostic approaches. And one of the things we are doing is defining pharmacokinetic and dynamic doses that can be used for clinical translation. So there is a lot to do. I think one of the questions is, how can we use this technology to improve efficacy in patients? How can we use it to better select patients? Dosimetry, also we have advantage of protein engineering now, so we can adapt the antibody to the biological question that we have. A total body PET that enables this sensitivity and reduced radiation dose. The multimodal and multiplexed approaches, and tumors being them so complex, how can we target multiple receptors in these tumors? And also, how can we take advantage of these AI-derived imaging biomarkers to help in guiding selecting tumors for therapies and monitoring therapy? And also, collaborations, dissemination, and this is a good platform to also work towards these directions. And I'd like to thank all the funding agencies, and especially the members of my lab. So Key and Sheila are two technicians in my lab. Key is exploring different statins in preclinical models. Sheila is working on defining drugs that we can use to enhance dotatate uptake and therapy in tumors. I wasn't able to show here today. Sandeep in my lab led the work on the metformin, and is also working on a really cool project of enabling uptake of multiple targets in the same tumor tissue. And Nina in the lab, another postdoc, she's mostly focused on the PD-L1 work that I showed in the last slides. And so with that, I thank you again for listening, and I think there is time for questions.
Oh, yes, that was a great talk. And I now request the audience, you can raise your hand for questions, or you can post it in the chat box so that we can get them clarified from the speaker.
Yeah, great.
Maybe I'll, yeah, great. Yes, Amol.
Yeah, I think there's a comment by Dr. Halker in the chat session. But before that, I would like to thank everybody for joining, especially we have people like Dr. Gasani here as well. So thanks, Munir, for joining. Thanks, Dr. Halker. So I would just like to read out Dr. Halker's comment that, very promising prospects in this great talk. 15 years ago, OncoSynth for colon cancer, ProstaSynth for prostate, for SPECT imaging using antibodies, and therapy with Bexer and Zevalin were tried. Maybe at that time, the timing was not right and awareness was lacking, but now with PET and beta and alpha therapy, we will succeed. So I would like to, expand on that. And one of the things I think at that time was we did not have better technology, and SPECT was not as robust with the advent of PET and the newer probes, as well as newer long-acting PET radiopharmaceuticals. I think we are in a better shape. Our oncology colleagues have always argued that they've been doing theranostics with with these receptor-based treatments. And my argument to them was, yes, but we don't biopsy all those lesions. So as you rightly showed that one of the patients was HER2 negative, but when they did the scan, a few lesions were positive and the patient was enrolled in the trial. We have to be careful in not overselling our technology either. So it's a complementary role. Like we have limitations in detection with PET. Granted that small animal PET has better resolution than clinical scanners, but generally speaking, we have about 6 to 8 millimeter lesions, especially if the signal to noise is good. But we will miss microscopic disease, micrometastasis, or low volume disease and low grade disease, depending on what we're targeting. But at the same time, biopsy is at the mercy of what was sampled. And if there are 10 lesions and you sample only one and label the patient as ER positive or HER2 positive based on just one lesion, we know that there's a lot of receptor heterogeneity, and we have to be careful. And that's one of the reasons when you say that a lot of patients don't respond or develop resistance, is probably because a fair number of those lesions may be not expressing the tumor. So we need to make our colleagues aware that this and I think a lot of your data should be published or people need to be made aware of that there's so much heterogeneity in the expression, and you need to complement tissue sampling with PET, and even we need studies to show that, okay, what happens if suppose there are 10
Light up enough.
Hypothetically
Light up enough.
Please, I think that somebody's
Please mute yourself if you're not asking any questions.
So hypothetically, if there are 10 lesions and say 8 are positive for the receptor of interest and 2 are not, what's the best therapy? Should we still go ahead with the anti-receptor therapy and treat the 2 lesions locally with, say, radiation therapy or something? And what happens is 50-50 kind of a thing. So those are the things that I think need to be done. And so people like you are involved in basic science research need to talk more with the oncologists doing the basic science research on that, and have a better collaborative approach, so that we can really say that we have a big stake in this personalized treatment approach in the 21st century.
Thank you, Dr. Amol. I think Dr. Halkar has a comment too. Interesting to note that metformin had negative impact in HER2. However, there is data that metformin helps in the treatment of head and neck cancer. Dr. Patrícia?
Yes. I think the metformin per se will also have some anti-cancer effect. I mean, we see reduction in HER2 signaling just with the metformin. But at the same time is inducing this increased internalization on the HER2 receptor. Again, what we see is that the effect is temporal. I know there was also a trial on metformin and testing in combination with trastuzumab, and on that trial they didn't see measure differences. So in terms of the therapy, again, I think the effect will be dose dependent, and also it will be very temporal. So, what we see in our preclinical models is that maybe these the fact that patients might be taking statins or metformin, maybe, when they receive a PET scan or when therapies are being defined for them, maybe attention should be made. It should be, people should be paying attention on whether or not they are taking these drugs.
Thank you, Dr. Patrícia. Any more questions?
Just a real quick one, taking advantage of your extensive knowledge, Patrícia. First of all, excellent talk, Mark Tulchinsky from Penn State, and then really very informative. But with all your wealth of knowledge, maybe you could inform us on the common observation that we all have made on FDG PET CTs. With patients on metformin, they typically have an intense uptake and mostly in the colon, but sometimes small bowel as well, but colonic uptake of FDG is quite intense. With your observations and the deep knowledge of the biology, do you have any reasonable explanation why that is? It's pretty consistent.
Well, metformin is like the statins. They have pleiotropic effects, right? Like statins are not only depleting cholesterol, they have many other effects in the biological system. Same thing with metformin. Even metformin, in the setting of aging, people repropose metformin in that setting. So maybe some of these pleiotropic effects, we didn't look. Actually, it was a comment from. We have this paper in submission, and we're going through revisions. And one of the reviewers asked that same question, do we see differences with FDG in our models? We didn't go into that direction. We had a focus more on the HER2 receptor, but I would think is mostly because of these pleiotropic effects that these drugs have. And for example, with in our study, because we use a mouse system and trastuzumab does not bind the murine HER2, we don't know if metformin is inducing changes in the other non-tumor tissues that we could visualize with the trastuzumab radiolabeled with zirconium-89. So it might be that these drugs are having other effects in addition to the effects that we see in the tumor cells. And so I don't have like an exact answer to why, there are those uptakes when patients receive a PET scan with FDG. I talk about this work with Dr. Wall. I think he has a paper, maybe, I think it's GNM, FDG and metformin. And he also mentioned that to me, that there are these changes in FDG uptake in patients receiving metformin.
Yeah, if it's better understood, hopefully in patients with colorectal cancers, you get such an intense uptake, you can't tell much as to whether it's, there is some pathological lesion or not. If that could be blocked, boy, I think a lot of physicians on this Zoom would be eternally grateful to you if you could somehow block that phenomena. But thank you so much once again. A wonderful presentation, very informative.
All right. Yeah. Thank you for your question. I'm gonna read more into that. And okay. Thank you. Thank you for the comment and for the question.
Uma, you're mute.
Sorry. I think Dr. Holka has a comment. Amol, can you read it out for me? Because I think,
sure. So Dr. Halker says he would like to know the resources you have at your institution, cyclotron, basic science labs, animal facility, etc. Do you get zirconium-89 from vendors or produce on site? If the production is on site, then what is the MeV of the cyclotron needed for it? Those kinds of details.
So the resources here are excellent, and that was one of the reasons why I moved here. There is also, I will go into the specifics, but there is also a very strong interest from physicians and radiologists, because I work on the basic side of things. We do a lot of preclinical studies, but there is such a huge interest from physicians and radiologists in what we do. So I have a strong collaboration with oncologists, breast cancer, gastric cancer specifically, and then radiologists like Dr. Farokh Dadashti. She's very well known in our field. And so I really needed those to be able to have this program that I want for my lab. In addition to that, the resources. So we have a really good cyclotron. They produce zirconium 89 every 2 weeks. They produce copper-64 every week. So these are the ones I use the most in the lab, but they also produce many others. The copper-64 is really good. They produce it to a lot of different places in the US, including MSKCC. The MSKCC gets copper-64 from here. Facilities, really good facilities. I can comment on the preclinical imaging facilities. We have 2 PET scans just for the suite that we use, the Avion, the Mediso. We also have PET MRI, SPECT. They're now working towards putting together a PET imaging facility for infectious disease, which I think is really cool and really needed. So yeah, overall, really good resources, cyclotron preclinical imaging facility, but also the collaboration and the Simon Cancer Center that we have here. The Cancer Center is also outstanding here, and that's why I moved here. Awesome.
So those excellent, yeah, it looks like you have a great setup. So those scanners are dedicated research scanners, or you share with clinical people, or how does it work? Especially, PET MR is an expensive scanner. So I'm curious if you have a dedicated research PET MR. I'm assuming it's a clinical PET MR, not a small animal PET MR, correct?
We have both. So we have the ones I mentioned, they are all in the preclinical imaging facility.
Okay.
That is directed by Dr. Koresh Shogi and Dr. Richard LaForest. And then there is the imaging facility that is more with Dr. Pam Woodard. So we have to separate things.
Excellent.
I haven't seen all the things we have for the clinical equipment or for the preclinical imaging facility, yes. And then there are all of the other equipments for IVs and MSOT and all of these other things that we don't use the most, but they are also there.
Yeah, I mean, we at Emory also have a new HSRB2 building with dedicated CTs and all that for clinical use. So in the scanners in the setting as well. I don't think we have a PET MR that is just for research. We have a PET MR, but it's 50/50. Like, it looks like really nice facility.
Absolutely. I think we are just in time. If there is any other question, anyone has another question? I do see a lot of young students. Well, I'm sure that your talk has been an eye-opener to many of them. And it was a superb talk. And maybe in the absence of any further question, I would like to thank the speaker and all of those who have actually joined it and had participated in the discussions. And for those who joined in, and I'm sure you would have taken home the message that Patricia gave in terms of imaging and in terms of the way forward with the various modalities of treatment of cancer for the diagnosis and imaging of them. Thank you, Patricia. It was really superb talk. And I think this is one of the first basic science talks that we have heard in this year's series of webinars. And hopefully there would be many such ones which can trigger the inquisitivity among the youngsters. Thank you. Thank you on behalf of all of us here.
No, thank you. And thank you for the invitation and for all the discussion and suggestions.
Patricia, special thanks from me, Jyotsna.
Thank you so much. Thank you for the invitation. And if there are any other questions, feel free to also email. I'm happy to answer the other questions. And well, have a good weekend. And thank you. Thanks.
Thank you. Bye-bye.
Bye.
Bye.